Sorry, I should have done that first. All right, great. So, Dr. I'll reintroduce you. So I'm recording now. So we're pleased to have with us today Dr. Nikhil Sahajpal, who's a fellow at Greenwood Genetics Center. Um, he um, he and I ran into each other a couple times at Cancer Genomics Consortium and have a shared interest in structural variants. He has done extensive work with optical genome mapping, and so he's going to share that expertise with us today. So thanks again for being with us, and go ahead and get started when you're ready.
Thank you, Beth. Thank you for the invitation. Let me share my screen. And you all can hear me fine, right?
Yes, I can hear you.
Are we in the right view?
Yes, you are. Looks
All
great.
right. Well, thank you. It's my pleasure to talk to you all today about the application of optical genome mapping in hematological malignancies. And for the discussion today, I'll be just touching upon the technology overview, and we'll touch upon the validation, but would like to share certain interesting cases that we saw during our validation studies, and then move into a bigger discussion with what might be the clinical utility of optical genome mapping, and does it have a potential to alter the clinical outcomes in these patients. And then we'll have a focused discussion on homologous recombination deficiency as one of its application. So the technology primarily works on isolation of high molecular weight DNA. And in this respect, it can be from peripheral blood, bone marrow aspirates, or solid tumors. Once the long DNA molecules are isolated, these DNA molecules are labeled at specific 6 base pair sequence motif. So this 6 base pair sequence motif repeats itself throughout the genome with a frequency of 4 kilobase pairs throughout the genome. So once this DNA molecule is labeled using fluorophores at this sequence motif, the DNA molecules are loaded into the instrument where they sit in a chip where the DNA molecule passes through the nanochannel arrays. So it first passes through microchannels, and with the help of electrophoresis, these DNA molecules are linearized. And from microchannels, it passes through nanochannels, where these DNA molecules are now running at a single molecule resolution. So at a single molecule resolution, the instrument captures images. And what you can see here is the blue lines represent one unique single DNA molecule. So the background is stained blue, and you might be able to notice the green dots, which are these fluorophores or the 6 base pair motif. So these images are then converted into digital barcodes. And the digital barcode, since you know the sequence or where these repeats occur, can be mapped back to a reference genome. So this is the alignment for these molecules. Once the primary alignment has been done, there is structural variation calling. And throughout this presentation and in this image, wherever you see a green bar, it represents the reference chromosome. The blue bar will represent the patient sample. And these lines that you see are the 6 base pair sequence motif that was fluorescently tagged. So when there's a reduction in distance between two labels, or there is a missing label, it is called out as a deletion. Similarly, when the distance between two labels increases and there's an additional label, it is called out as an insertion. When the labels repeat themselves, you can actually count the number of repeats, and the software does that for you. So you can capture repeat expansions and contractions. When the labels simply repeat themselves, you can check the orientation of these repeats, and can determine if it's a tandem duplication or an inverted one. And in this case, it's a more complicated rearrangement where there's a segment in between, but looking at the molecules, you can see that this is a tandem duplication. When at instances, for example, here, the labels in part align to one chromosome and the other part aligns to the other chromosome, it is called out as a translocation. And if the labels simply flip themselves, it is called out as an inversion. So it's a play of how the labels orient to call out different structural variation classes. So the data that I'm showing is primarily from my previous lab at Augusta University, and where I'm doing my fellowship right now, we have active collaboration between the two institutes, between Greenwood Genetics Center and Augusta University. So we had validated optical genome mapping for different applications, postnatal, prenatal, and oncology, both hematological malignancies and solid tumors, but I'll just focus on the heme part here. So initially we had done a validation in hematological malignancies, and what I want to point out here is that we included different tumor types ranging from AML, CLL, MDS, plasma cell myeloma, lymphomas, and CML. We included the different sample types that we see in our lab, such as bone marrow aspirates, peripheral blood, CD138 isolated cells, and lymph node single cell suspensions. We did different stability studies, which I'll not go into. But most importantly, most of these tumor types or the samples were analyzed in the standard of care workflow with karyotyping and FISH. While we included different classes of SPs ranging from aneuploidies, deletion, duplication, translocations, isochromosomes, inversion, ring chromosomes to capture the plethora of structural variations that you'll see in these tumor types. And not going into details, just to highlight the overall results of the validation study, the optical genome mapping showed an overall sensitivity of 98.7 and a specificity of 100%, while you can see the sensitivity and specificity and the other precision markers for different SV classes. So overall, compared to the standard of care workflow, optical genome mapping showed high sensitivity and specificity to pick up those rearrangements and the different classes of SVs. One experiment that I do want to highlight is the detection or the lower limit of detection with optical genome mapping. So we had analyzed these genomes at a depth of 320x. And what you see here is 4 different SV classes: translocation, an interstitial deletion, interstitial duplication, and an aneuploidy. And we diluted the samples from 50% allele fraction, which was diluted down to 5% allele fraction. And what you can notice is that the technology was able to pick up these different SV classes down to 5% VAF at 320x depth. I do want to highlight certain interesting cases that we observed during our validation study, where with the higher resolution, we were able to correctly interpret what was being seen with karyotyping and FISH. So the first example in this series is an example of a CLL case where we saw this complex karyotype, but I do want to highlight chromosome 21 here, where we see an additional 21 and with dicentric 21-21. So to confirm this, we had run FISH with RUNX1 probe, and what we can see is the RUNX1 is the green probe, and we can see 3 signals for RUNX1. So seeing this chromosome structure and 3 copies of RUNX1, we did finally interpret this as a +21 dicentric 21-21. But when we ran this sample on optical genome mapping, just to run through how this is interpreted. So if you look at the optical genome mapping data, the first view that you see is this circus plot. And the way it runs is chromosome 1 through 22, X and Y, and you can see the cytobands. The smaller copy number changes or structural rearrangements are highlighted in this ring, whereas the large copy number gains or losses are highlighted in this copy number track. So you can see while for autosomes it's at copy number state of 2, and for X and Y it's at 1, so it's 46XY. You can see that there's no copy number gain on 21, which should have been 3 copies if it was 3 copies of 21. But we can see there's a translocation from 21 to 1. And if you zoom in, you can see that this translocation actually breaks right in between RUNX1 gene. So what's really happening is that there's a portion of chromosome 21 going to chromosome 1. And that's the reason that split the RUNX1 probe into 2, and we see 3 signals for the RUNX1 probe. So it's missing these subtle rearrangements, and the higher resolution can help us interpret these genomes better. So the second example that I would like to highlight from the validation study was a case of plasma cell myeloma, where we saw 3 red signals. The red here is FGFR3, and this was interpreted as a gain of FGFR3. Although I do want to highlight that the tech that looked at these fish cells said that noted that the third signal is smaller, so this might be a rearrangement of FGFR3. While if we look at optical genome mapping, what we can see is that there's no copy number gain, while there's a translocation which misses the FGFR3, and the breakpoint is in the gene which is distal to it, the NSD2 gene. So both of the interpretations, either a gain or a rearrangement of FGFR3, are in a sense incorrect, because the FGFR3 gene is neither amplified or rearranged. But just to probe this case further, this plasma cell myeloma case was reported with a normal karyotype and 3 changes, alterations with the fish probe. When we ran the bone marrow aspirate on which the karyotyping was done, you can see that while the karyotyping was normal, you can see several rearrangements with optical genome mapping, which highlights that it has the ability to pick variants which are at a low variant allele fraction. But what would be of more interest here is that we were able to isolate DNA from the CD138 isolated cells. And you can see that the picture here is much more complex than what we get with fish. So we can see the gain at 1Q, the 4p-FGFR3 gain, or the rearrangement here seen with this translocation. And we can see the monosomy 13, which was picked by fish, but we can see the genome is much more complex. All right, so I'll end there about the validation study, and if you want to read more, you can read with the paper that was published in JMD. But I think we and other groups at that time, which were in the same boat of validating the technology, was asking this question after realizing that the technology does have great potential to lead to several insights into these tumors, but how can we leverage the current guidelines, and does it have the potential to alter the clinical outcomes in these patients? So we recently did a small exercise where we looked at how many cases do we have in our lab which have at least 2 years of clinical follow-up. So we looked at the cases that were run in 2020-21, and out of the 75 cases, we have 48 cases which were run, which have 2 years of clinical follow-up data. Of these 48 cases, 18 are AML cases, 10 are MDS cases, and 20 are others. I'll not talk about the others for now. So we did ELN risk stratification for the AML cases, and compared the risk stratification with standard of care with optical genome mapping. For the MDS cases, we did IPSSR risk stratification, compared SOC with OGM, and then followed these patients and measured clinical outcome as a measure of survival. Now, before I go into that, 28 of these cases did fell into another bucket, which is we proposed an altered workflow for myeloid cases, since I'm talking about AML and MDS here. So what we have been traditionally doing with myeloid cancer samples is that we divide the sample into two parts. One goes into the molecular lab, one comes into the cyto lab. In the molecular lab, we do targeted NGS panel, which range from 12 to 54, or some institutions use 80 gene panels. We use PCR to capture FLT3 ITDs, TKDs. And in the cyto lab, we use karyotyping and FISH, and both the molecular and the cyto report are reported out separately. So we had proposed that in the molecular lab, instead of a smaller gene panel, we use a 500 gene panel. We'll still have to use polymerase chain reaction for FLT3 ITDs, while in the cyto lab, instead of the karyotyping and FISH, we use optical genome mapping. And then we can do a combined or a combinatorial analysis with the 523 gene panel and the OGM in a single software and report as a single report. I'm highlighting this because I'll be talking about certain cases when I show the outcome data. Just to highlight the key result, and I don't want to emphasize on any of these results, but just to highlight the combined analysis, that in the 30 samples that we analyzed using this method, we identified that there are 13 compound heterozygous events per case. And out of the 30 cases, we found potential clinically significant events in 12 cases. So that's one of the aberrations that I think we are missing with the current workflow. All right, coming back to the outcome data, what you see here is the risk stratification with karyotyping and FISH. And what I have done here is that I combined the favorable with very good and good from ELN and IPSSR. Intermediate remains intermediate, and the adverse in ELN risk stratification is combined with very poor and poor with IPSSR. But when I talk about these cases, I'll specifically mention which are MDS and which are AML. So what you can see here is that two of the MDS cases, which were very good with IPSSR, were changed to very poor with optical genome mapping, and one AML case, which was stratified as intermediate risk, was reclassified as adverse with optical genome mapping. And when we follow these patients for the next 2 years, what you can see here is that the patients that were reclassified into the very poor or adverse group did had a clinical outcome that correlated with this risk stratification. While we had one outlier here, an AML case, which was favorable with both these technologies, had a RUNX1 RUNX-T1 translocation, but on a follow-up marrow had adverse profile with both these technologies. So just probably the way the tumor progresses. So I want to highlight just one case, one MDS case, which was changed from very good to very poor. So if we look at this case, a 60-year-old female, 1% blast, the sequencing was negative with the 54 gene panel, and with karyotyping had a translocation X12 and a deletion on the long arm of chromosome 20. So a good prognosis with IPSSR risk scoring. Now the patient was put on chemotherapy, and a 16-month follow-up marrow showed a typical normal karyotype 46XX, and the patient died 18 days after this follow-up marrow. So if we look at the case at time point 1 with optical genome mapping, what we can see here is that in addition to the X12 translocation and the deletion on 20, there are several other cryptic rearrangements, probably at a low variant allele fraction, which were not captured. And if we highlight into the chromosome 13 region, what we can see here is that there are multiple clones. A clone that deletes DLU1, another clone that deletes RB1 in addition to DLU1, and there are several translocations at these within this region. So overall, to say that it was a very poor risk stratification with optical genome mapping. So just to say, if the physician was aware that the risk stratification group is very poor and the profile is complex, would there have been an altered therapy that would have been pursued at this time point? But to go on with the proposed workflow that we had suggested with 523 gene panel and optical genome mapping, I would like to highlight that a small structural rearrangement here on chromosome 11, where we see that there's a small deletion that deletes MRE11 gene. And with the sequencing panel, we can see there was a nonsense variant in MRE11 gene, which was classified as a tier 2 variant. So a biallelic loss of MRE11. And how this is relevant, since we'll go into homologous recombination deficiency in a moment, MRE11 is a primary protein in the initiator complex of the HR pathway. So another, I'll open up this for discussion when we are closing in on this presentation. But if we look at even the 16-month follow-up marrow, where we saw a 46XX with karyotyping, what we can see here is that there are smaller cryptic rearrangements with optical genome mapping, including loss of ETV6 and EZH2, which have an independent poor prognosis. So just to say that we missed out probably relevant information at both time points, which the clinician and the family probably deserve to know and could have made an altered decision with respect to therapy that they pursued. So summarizing the part 1 of the talk here, that the risk stratification appears to be more accurate with optical genome mapping, while the comprehensive workflow with optical genome mapping and the 523 NGS panel has the potential to provide clinically relevant and potentially actionable variants. And I do believe that in the coming years, that the risk stratification guidelines and the diagnostic criteria would be altered, and the OGM data would play a critical role in these modifications. So coming to what might be of greater interest to this group, homologous recombination deficiency. And we had asked this question that, are we using the right tools to calculate homologous recombination deficiency scores? And we'll come to why this question might be relevant. But just to briefly review what is homologous recombination deficiency. So there are two major pathways in our cells to repair DNA double-stranded breaks. One is the high-fidelity, error-free homologous recombination repair pathway, and the other is non-homologous end joining, which is rather error-prone. So when these pathways do not function as they are supposed to, this results in additional aberrations, which results in unregulated cell division and cancer susceptibility or progression. Why this is relevant to pick up homologous recombination deficiency as a simple example here is that when we have DNA double-stranded breaks, there are two key players, BRCA and PARP, which are both able to repair these double-stranded breaks. In cells which are BRCA deficient or have mutations in BRCA gene, the PARP is able to take up this DNA repair function, while what can happen is, if you can recognize HR deficiency or BRCA deficiency here, you can inhibit PARP, which will result in additional accumulation of double-stranded breaks, and the cell will undergo apoptosis and cell death. So this is the mechanism that is leveraged in HR deficient cells or tumors. Now, it's not that simple. The HR deficiency is not just caused by BRCA1/2 mutations, but there are other HRR genes, and at least 24 have been recognized that alteration in those genes results in HR deficiency. And there's not just sequence or copy number changes, but there are epigenetic events that might result in deficiency. And then there are yet unknown reasons that might result in HR deficiency. So one way could be that we capture all these alterations, but is obviously complicated because it's not just one mechanism, not just sequence DNA changes, but epigenetic mutation, yet unknown causes. And the other is that once there is an HR deficiency, which can be due to any one of these reasons, it results in a genomic instability, which has a characteristic signature. So you can capture this genomic instability or the signature, which can represent the HR deficient tumor. So all this to say that if you capture that this tumor is HR deficient, these tumors are sensitive to PARP inhibitors or platinum-based therapies. So we'll primarily talk about this genomic instability and capturing these signatures. These signatures are also called as HRD scars, and there are 3 different scars that have been found to be characteristic of HR deficient tumors. The first is loss of heterozygosity. The second is telomeric allelic imbalance. And the third is large-scale transitions. Now, I would like to highlight that all these 3 scars were identified using chromosomal microarray as a technology back in 2012, and we'll review how these were identified. It is interesting that all 3 were identified in the same year in 2012. The first group published this report in epithelial ovarian cancers, where they identified that loss of heterozygosity predicts HR deficiency. And how they defined HR deficiency is these number of regions which might be copy neutral or copy loss and are greater than 15 MB in length, but do not include the entire chromosome. So you count those number of loss of heterozygosity regions, and they had a cutoff above which it would say that this tumor is HR deficient or HR proficient. Then in the same year, another group published telomeric allelic imbalance that correlated with HR deficiency. So how this group defined telomeric allelic imbalance is the number of regions which might be copy number gains, loss, or LOH that extend into the subtelomere but do not cross the centromere of that chromosome. And these events are subject to debate. Some groups have used 3 MB as a criteria, that the event should be 3 MB or greater. Some have kept at 10 MB. And the last scar that was identified in the same year was large-scale transition, where the large-scale transition was defined as chromosomal breaks that happen in the genome due to events that are 10 MB or larger. So these can be copy number gains, losses, or LOH. And there are certain additional rules which we can talk about in a moment. But more recently, what has been shown is that a combination of these 3 scars, if you add these 3 up, are a more sensitive measure of measuring HR deficiency or proficiency as compared to one of these scars alone. So when we discuss about HRD, we'll include all these 3 scars to see if what has been done and how to calculate these scars. All right, so example 1 here of an LOH. So what you see here is a copy number loss, which is 17 MB in length. So given that this is greater than 15 MB, this becomes an LOH event and gets a score of 1, due to that LOH event, but it additionally results in chromosomal breaks at these two ends. So one score for each of these breakage. So this is characterized as LOH with a score of 2 chromosome arm breaks. So this event alone has a score of 3. Then the next is a TAI, where you can see there's a copy number loss which goes into the telomere but is not extended into the centromere. So because it extends into the telomere, it is a TAI. Now the length of this TAI is greater than 15 MB. So it qualifies both as a TAI and also as an LOH. So it gets 1 score for TAI, 1 score for LOH, and because there's this chromosomal breakage, it gets 1 score of LST. So a total score of 3. Now, given if this copy number loss was less than 15 MB, then it would not have qualified as an LOH, but only as a TI and would have a score of 2. Coming back to the same example, LSTs are chromosomal breakage where the event is greater than 10 MB. Given that this is greater than 10 MB, it counts both these breakages, so a score of 2, plus 1 score for the LOH. Now, HRD has been well studied as a marker in ovarian, prostate, breast, and pancreatic cancers. The more recent papers use all 3 scars to calculate these HRD proficient or deficient tumors. Would want to highlight that we have been using NX Clinical as a software to pick up these scars because it has a script and an automated tool that can capture the HRD scar, and we'll show you a few examples of that. But I do want to highlight that although that these 4 tumors have been well studied and the HRD phenotype characterized, the scores have not been able to be found sensitive in other tumor types. And even if we talk about ovarian, breast, prostate cancers, this is a recent ESMO recommendation. And if I can highlight just the abstract here, what you can see here, the group saying is that these tests, and when they talk about these tests, they are talking about chromosomal microarray or sequencing to pick up copy number changes, that their use is limited by failure to consistently identify a subgroup of patients, and they lack a negative predictive value. So while on one hand, they are able to have a good sensitivity with respect to positive predictive value, but they certainly miss a subset of patients even in these tumors. And we can discuss why that might be a reason in the way those clinical trials were conducted. But this point and the fact that HRD as a biomarker has not been able to found to be that sensitive in other tumor types does pave way for other technologies or other ways to capture these deficiencies in these tumors. So we hypothesized that what has been missing in these scars is certain rearrangements, balanced or cryptic rearrangements, which are also DNA double-stranded breaks, but are not captured by the technologies that we are using. So here is an example of a glioblastoma case where you can see these copy number changes and amplifications, but this tumor is enriched by several rearrangements, and all these lines represents rearrangements between these chromosomes. And when we look at different tumor types, for example, here glioblastoma, lymphomas, and uterine tumors, they are all enriched with these rearrangements, which we have been consistently missing in our SCAR or HRD score calculations. So what we did is we looked at certain tumor samples with all these 3 technologies: chromosomal microarray, a 500 gene panel which can do copy number variant calculation, and optical genome mapping. So I'll start with a case of glioblastoma here and compare chromosomal microarray with optical genome mapping. This is an example of glioblastoma where you can see there's a copy number loss on chromosome 6, a copy number loss, an interstitial loss on chromosome 9, and a terminal loss on 19. And this is a picture from NX Clinical, where you can see it calculates the scar calculation once you refine those calls. And the overall score here is 7. And it splits how it's giving those score to you. When we ran the same sample on optical genome mapping, you can see that it's much more complex. And the scarring is driven or is more represented by these rearrangements rather than those copy number rearrangements or those transitions. And the score here is 32 on the same sample. So just to look at the calls with both technologies, you can see how these two technologies are concordant with respect to copy number calling, where we see this interstitial deletion, a region of greater than 15 MB, so an LOH score 1 and 2 scores for LST, so a total score of 3 for this event. We can see the terminal loss with both these technologies, a TAI event, and then we can see a LOH event. I do want to highlight here, this is an event where you can see that there's a terminal loss which actually goes beyond the centromere. So it's no longer a TI, it's only an LOH with 1 break, so a score of true. So there are certain subtle nuances to how the scores are calculated, but this is the way it was done in the initial clinical trials. But if you look at the higher resolution of optical genome mapping in these tumors, I do want to highlight here that while we see with both technologies, there's a copy number loss and interstitial deletion here, which is 5.3 MB in size. So it does not meet any criteria, does not meet the criteria for LOH, does not meet for LST, because for LST, it has to be more than 10 MB. But if we zoom into this event further, what you can see here is that this event actually results because of 2 translocations. So there are 2 LST events, and in order to calculate LST events, there's further nuance that the 2 events need to be 3 MB apart. Since this is 5.3 MB region, these 2 rearrangements are 3 MB apart and contributes to a score of 2, whereas it was a no score with CMA. We have extended this to myeloid neoplasms, and I'll just show you one example here where there's this copy number loss of 17.2 MB. While it gives a score of 3 with these 2 technologies, there's an additional, you can see there's a yellow map here at one of these breakpoints, which goes into chromosome 11. So while there was no rearrangement on chromosome 11 with the NGS panel, we can see that it results in a breakage with optical genome mapping, so would add to additional scoring. So with the limited samples we have done, what we can observe that the increase in the score is not linear, that certain samples which have a low score with one of these technologies do have a greater score with the other one, and these might be the outlier cases if we can extend this experiment to the tumors which are established with HR deficient or proficient, such as breast and ovarian cancers. We also did a validation study where we generated a script for data coming out from optical genome mapping, and this is the automated score versus the expertly curated score, and we had refined this to a level which is acceptable for concordance between both of those methods to capture HRD phenotype. So to summarize this part, so there seems to be that optical genome mapping does have a potential to capture additional variants, and which might able to stratify further the HR proficient and deficient genomes. and we published a first draft of just the calculations. We did not have any of these tumors or patients in a clinical trial to measure what they're responding to the PARP inhibitors or not. But just highlighting this paper because we listed out all the rules that we had used to compute these scores. And also the script is submitted to a public domain that can be used to calculate HRD if you have optical genome mapping data. All right. I'll just take 2 minutes more and then I think we can go into the discussion. But I would like to highlight that when we see all this data and we had asked that what's next. So we at Augusta University did launch this for at least the hospital patients. And the first case that we had done in our institute on a prospective case for which a report was issued was this female, which had 90% blast, normal karyotype, normal fish, NGS report was negative. And these were the genes that were in the NGS panel. You can see the karyotype is negative, and there were several fish panels that were done on this patient. And when we ran this on optical genome mapping, you can see that we see a cryptic 719 translocation, and then a rearrangement in 10 and 11, which was actually very complex, where just this region of chromosome 10, which is MLLT10, was inserted in chromosome 11, breaking the KMT2A gene. So it formed the KMT2A MLLT rearrangement in a way that would also occur in classic translocations. So this is how the report looks like, and this is what the clinician had asked for, that what they want to see in the report. And just to conclude, I think we are in this stage of molecular cytogenetics that we certainly see a place for optical genome mapping in our clinical workflows. And would like to thank all the people that you see on this slide, but especially Dr. Kole, who is the director at the pathology lab in Augusta University, and where all this work was done, and who is continuing to use this technology for clinical reporting. Thank you.
Great, thanks so much. So we're open for questions. So anyone who has questions, feel free to ask them. I'll kind of start out. You mentioned that your HRD algorithm is publicly available. How would somebody access that?
Yeah, so we have given the link in the manuscript, and
Okay.
I can forward you the manuscript to you, but it was submitted to GitHub, so it's the script is available there.
In GitHub, perfect. Cool, thanks.
Hi. So very, very nice talk. Thank you very much. I have many questions, many discussions, but I would focus on a few. So you said you're using NX Clinical for the 523 gene panel is the TSO 500, correct?
Yes, yes.
Right. So have you tried both CNV validation, focusing on deletions, and HRD score with TSO 500 data, right? If I understood correctly.
Correct. Yes, so we did look at the TSO 500 data to capture copy number changes and compared it to OGM here. So I would say for HRD calculation, the TSO 500 data can be used because we are focusing on larger deletions and duplications, because the minimum that you use for your calculation is 10 MB. While at the telomeric allelic rearrangement, we have kept a threshold of 3 MB, but since the guidelines are not clear if it should be 10 MB or 3 MB, so we have kept 3 MB or above. So for HRD, TSO 500 data can be used if you're just focusing upon capturing copy number changes for HRD calculation.
With good sensitivity. I mean, capturing most cases that would be called HRD positive by other method, either array
By CMA.
or...
Yes.
So it's
With
good
a good
enough.
sensitivity. Yes.
So good. That's a question I've been trying to answer for a long time. So one more question, because there are more people interested in questions. Do you know of any comparison of whole genome sequencing with optical genome mapping? I mean, the sensitivity for detecting all these complex rearrangements and smaller copy number events.
Yeah, so a head-to-head comparison in oncology has not been done with whole genome sequencing and optical genome mapping. But there's certainly that paper from in NEGM from the WashU group with whole genome sequencing. And there have been papers on the optical genome mapping side, but both of them compare to the standard of care karyotyping FISH or CMA, but there has not been a comparison of OGM and whole genome sequencing on the same set of samples. Although when we proposed the altered workflow with 523 gene NGS panel and OGM, we did theoretically compare this workflow with the whole genome sequencing, and also compared theoretically in discussion. There's a section on the discussion section comparing the OGM and 523 with whole genome sequencing, but there has been no head-to-head comparisons in the same samples. That can answer.
Thank you very much. Congratulations for the work.
Thank you. I think Moshimi, you had your hand up first.
Yeah. Yes. Hi, Nikhil. Very nice presentation. So I have some first basic questions. So you need fresh tissue or fresh sample only?
Correct.
Formalin fixed.
No, the FFP doesn't work. So
Ah.
you need fresh or fresh frozen, which was
Fresh
frozen when it was fresh. Yes.
Yes. Okay. Another thing is, what I can see here is a technology which is more sensitive, or which appears to be better than the previous technologies. So when you are validating, so does it actually make sense, or to compare it with the gold standard, which actually appears to be far lesser than the
Correct.
technology itself?
Correct. So,
But then,
so we and others had faced a lot of problems during validation, and had several comments from the reviewers that what would be a good measure. But what we have done is that when you capture the clinical sensitivity or the positive predictive value, we compare it to karyotyping in FISH, that what was seen by the standard of care, can we see that? And for the additional variants, now there are two options, that you validate all those variants, which seems very difficult. So we used healthy controls, and for true negatives, we use those healthy controls to calculate the true negative. So you should not see anything in those genomes. And if you do see, then you need to validate that if it was true or not. But that would be the only way to capture the true negative value. And for the labs that have clinically validated it, we came under a common umbrella with the US, Canadian, and European groups, and formed a consortium, and wrote a framework paper where we suggested all those, what can be used to calculate true positive, true negatives, and how labs can validate that. And that paper should be coming out shortly this month or the next. But it's a very complicated question, because obviously the standard of care does not have that resolution, and you see a lot more. So what remains true positive and true negative is certainly need to be defined before you start validating the technology, that what your criteria are.
So were there any cases which were positive on fish or karyotype and negative on this, like which did not? So did you have those cases?
So we were too naive. I think when we started, we included the additional materials or ads also to compare, and we had an ad which did not make sense. We did not see that ad, and we could not make sense using the OGM. So we called it as a true negative finding. So that was one event that was missed. And in the previous version of the software, there was we missed one copy number loss, which was picked up by, I think, karyotyping on either Y chromosome or X, which we did not see it, but we reran that with a new software and we can see that change. So I think it has improved with improvements in the software, but I cannot recall where something was picked by karyotyping in fish and we missed it.
Okay,
And
and
it-
another thing.
Oh, sorry, I
Go ahead.
was going to jump in here just really quick because I have a little bit of experience with a similar technology, and we would miss some copy number variants and translocations that had breakpoints occurring in repetitive regions. They ended up being masked by our technology. And I think that's kind of the downfall to a lot of these molecular techniques. I'm wondering if that might be a similar reasoning behind kind of like missing a CNV loss.
No, so the one I was talking about was a whole chromosome what was at a low VAF. The software didn't call it in the previous versions, but it does call it now. But
Got it.
do want to highlight the repetitive regions and those segdupes where with the molecular techniques, the rearrangements can be lost. And that might be one comparison that needs to be done with the genome sequencing and OGM. But I would say that with the current software and the reference genome that we are using, if the breakpoint occurs in the centromere, then we would miss those rearrangements because there are no labels in the centromere, or the sequence is not known with the HG38 or 19 reference that you use. So we haven't explored the T2T yet, and that would be something that remains to be seen. But certainly if the rearrangement is happening in the centromere, or one of the breakpoints is in the centromere, then you would miss that rearrangement.
And another thing about HRD. So what I could understand is that all HRDs on NGS or microarray, which are positive, are actually positive, but the low ones, okay, the low ones were positive on optical genome mapping. There were few which were low, which were actually positive. So if you have a clear-cut positive, so you can take it as positive, but a low one maybe needs a further testing if to exclude that it might be high. So how many of the low ones were actually falling in that level, or was there a cutoff like below which was different from the usual cutoffs? Because even usual cutoffs are not very standardized, like different panels have different cutoffs and you don't really know exactly what the truth is.
Correct. So I think I failed to mention that we did in brain tumors and myeloids. So those were just for estimating how do the scores compare between these technologies, and is there a linear increase or not, which we did not see. But these patients were not under clinical trial or with the PARP therapy that we could see the response, they are responders or not. But the problem with respect to HRD in the ovarian or breast cancers is that the way those clinical trials were done was that you estimate their HRD and those patients are on PARP inhibitors. So you can see that they are responding or not. So the positive predictive value is 90%. But those that, for example, now, if you have a cutoff in breast cancer, let's say 44 score and above, so those will get the PARP inhibitors. But those that do not have a score above 44, how many of those had an HR deficient tumor that do not get PARP inhibitors, right? So there are two groups, one that receive PARP inhibitors 44 and above, and out of those 90 respond. But there's another group that do not reach that threshold, are below it, who do not receive the therapy. So I think one is to address that 10% that do not respond, but then there's a bigger group that do not cross that threshold. How many of those patients do have an HR deficient tumor?
And another thing, does HRD work in tumors beyond the 4, like even if it is high, like beyond the prostate and the ovary and breast and pancreas? Does it actually? You have done it in GBM and myeloid. What is the reason? Why didn't you take the usual ones? Because and we are not sure that it works. I don't think it's been reported to work in these tumors.
Correct. So there are right now 150 plus clinical trials that are based on HRD, and there are all different criterias where there's scar calculation or there's IDH mutation. And there are clinical trials going on in myeloid tumors and gliomas and glioblastomas. But the criterias are all different, and it's not established what to use. We do not have patients in any of the clinical trials in any of the tumors. So all we did was compare the score calculation and put this out that those groups that are doing clinical trials can do a subset and see if which technology might be more sensitive.
Yeah.
Sounds
Thank you.
great. In the interest of time, I'd like to move on. I mean, I know we're at the top of the hour, and if you're okay with going a couple of minutes
Sure.
over, I'd like to give people who have had their hands up opportunity to ask their questions. So, Subit, I think you had your hand up.
Yeah,
next.
thanks, Nikhil. It was a great talk. I was wondering, I had several questions, but I'll limit to one. You mentioned about the Augusta University about this and your work. I was wondering, what is their workflow now? Are they clinically giving OGM as a primary test, or they're reflexing after karyotype and FISH as a reflex test, which is negative?
I think as of now, they are using the research money, and for a subset of patients, they give out both the karyotyping and FISH report and the OGM report. So,
and it is from the research. You're saying it is mainly on the research protocol they're providing,
yes,
right?
yeah,
Okay.
Using the research money, there's no reimbursement, I think.
Yeah, that's it's we all wonder about that part. Yeah, thank you. I'll stop there.
Sure.
Thank you so much. And I see we have another hand up, Dr. Simbu.
Hey, hi, thanks. So yeah, it's a very good presentation. Just I have 3 questions, but I will limit it to 1 question right now. So it's almost related to Dr. Somi mentioned earlier. I do see here that OGM technology is supreme to the karyotype and other thing. But one thing is clinically, right, how much it is relevant. So OGM can able to pick up a lot of new translocation and mutation which were missed out in the standard of care testing. But without validation, how much clinically it is relevant, right? Because the clinical change today, the challenge in today in the oncology segment is the mutations which we are going to identify in the OGM, even the well-validated foundation test or garden tests, all those things in the VUS category, variant of uncertain significance category, we do pick up a lot of mutation, but we cannot do anything about it. I do have very interesting patient who is suffering from ovarian cancer, and there is no actual mutation were picked up in both tissue and liquid. But if I looked into the VUS category, same mutation in the blood as well as in the tissue picked up. So we could not find out the actual driver since it is not well validated. So the doctor cannot take any clinical decision after that. So the technology is supreme. Yes, we agree. But on the other side of it, that how it is going to clinically helpful if the scientists like you are not validating it is clinically able to change the treatment plan or benefiting the patients in the clinical meaning way. So that's the one concern for me. Yes, as a scientist, I do have while I was working in Singapore, lot of mutation we picked up. Now today's world, the clean war have dumped with a lot of US mutations, but without clinically validating how it is going to benefit the patient or the clinician who is going to write this expensive test in the future for the patient actually.
So I think there are two tangents to this question. The first is how it is clinically relevant. And I think I try to include that part in the talk by showing one of those cases where it was a good prognosis with what we could see. And if we had known that it was not a favorable prognosis and altered therapy could have been pursued. And we and also other groups have shown that there are clinical trial opportunities when you capture those aberrations, and there's risk stratification changes. So only when we start using this in the clinical workflow, it would start impacting those the outcomes in those patients. And the second point, I do want to touch upon that, because I think we as a scientific and a clinical group do have the responsibility to capture those additional variants, because they are not in the guideline or in the stratification as of now because they were never captured. But once you see that those trends, those more might better predict the outcomes than what these stratification tools do right now. And once you see those recurrent rearrangements or fusions, I think fusions is one of those classes that have been missed for a very long time. And we have seen with what we already know that several of those fusions do drive tumors. So there are additional drivers that we have not been capturing, and only if we start capturing those, the other groups that are purely research based would go on and pick up on those and exploit if those are targetable targets or not. So I think even now the technology is relevant to how the clinical the therapy selection would alter, and there are several clinical trials. I think Rashmi's paper showed a few examples where they were able to pick rearrangements for which there's an active clinical trial going on right now, and there's obviously we have that responsibility to capture more and see what we can research upon.
Yeah,
Right.
totally. I agree with you. The chicken and egg story of identifying the VUS mutation, one side of it, and validating the VUS mutation. It's like chicken and egg story. Clinician and the scientists have to come together. I hope some of the funding will be more focused, at least in the oncology segments, to well validating the VUS category mutation to enrich the ClinVar database so that it will be meaningful when it is reporting in the reports. And another quick question, if time permits for me. So mainly it has been SOC kind of for testers have been utilized in these calculations or comparisons between the OGM. Is it like a well-validated testers like myeloid or any other testers which is for well-known for HRD testing have been utilized? How supreme OGM technology is more than these technologies? Because the technology is one part, but another side of it is software. Even in the NGS is also TSO 500 is good. Same use as the hybridization capture technology, but the bioinformatic parts, that is where the validation as well as some part of it is going to be different actually. So like is there any way that in the future, I understand that in this presentation it is not that in the future, that are you going to plan to utilize well validated testers in the NGS category as well as in the microarray category, like AMIDX and etc other stuff?
If I can ask, what do you mean by well validated? Just trying to understand.
Yeah, sure. So well validated in this case, like for an example, TSO 500 is not at like FDA approved already, right? So, but if you look at that testers of Foundation or Guardant and all those things are well validated actually, right? So that the PPA as well as the NPA values will be much robust, and also we can able to identify it, and it will be clinically meaningful. So if OGM can pick up when you are comparing with that kind of ingest technology rather than just with the TSO 500, and same goes for the HRD testing. So just normal CMA rather than normal CMA, if you are comparing with the commercially available technology which is validated and approved by the FDA technologies, right? So those kind of technology, if you are utilizing, then it would be a little bit more helpful for the clinicians per se.
Well, I'm aware of that. There's work that is being done to compare both TSO 500 and optical genome mapping with the those platforms such as Myriad and others.
Oh yes.
Mm-hmm.
That work is underway, but I do not have the
No worries.
the insights to
Those
say
are the-
anything on that.
Yeah.
That's okay. No worries. Thank you so much.
That's a really great conversation. Thank you so much for everybody participating in the discussion here. I had a feeling this was going to be a really great discussion, really great presentation, and you delivered. Thanks, Nikhil. I just want to expand a little bit on one point before we close out. I know that because of kind of my shared interest, our group has looked into, not just like looking at novel variants, but like you presented today, like novel profiles or new ways of like measuring genomes. So any way that we can like assign an attribute to different samples within a cancer type, it may become a way of measuring these tumors. And even though it may not be clinically helpful right now, maybe 5 years down the road, it will be. So really cool stuff. So thank you again for being here. Apologize for going well over our hour, but this was just such a good discussion. I did not want to hinder it. So thank you so much, everybody. Nikhil, are you okay with me distributing your email address for those who have additional questions?
Absolutely, yeah. And
Okay.
thank you for the invitation. I really enjoyed the conversation. I think I should have kept the talk a little.
It was perfect. It was perfect.
Okay.
Yeah, great. Awesome. Well, everybody have a great week. I'll see you in a few weeks. All right.
Thank you.
All right. Bye-bye.